One panel is the Western diet, and each one of these bars, the first bar is cerebral spinal fluid, and this is percent change from baseline now. The second bar is gray matter. The third bar is total brain volume. The fourth bar is white matter volume, and for Mediterranean and for Western. So just an immediate kind of a impression. What you see here is that all the brain volumes are changing radically from baseline in the group consuming a Western diet, and none of them changed significantly in the group that consumed the Mediterranean diet. So it looks like the Mediterranean diet is preserving brain structure again over this period of time, which is similar to a 9-year follow-up during middle age. In the AD literature, when we were doing this study, Schwartz and some other folks were calculating meta ROIs and both thickness and volume meta ROIs. And that represented all of these different brain areas, which are the ones that are impacted earliest and most significantly in Alzheimer's disease. So we did the same thing. We constructed a non-human primate Alzheimer's disease related thickness and volume meta ROIs. And that's what you see here. So this is the cortical thickness meta ROI in the Mediterranean diet on the top, Western diet on the bottom. And white is the biggest change, and you can see that the biggest changes are occurring in anterior left to posterior right. And what you can see is that the biggest changes that we're seeing are occurring in the Western diet animals, and they're positive changes because this cortical thickness is primarily gray matter, and gray matter increased on the Western diet. Well, that's really interesting because the ADNI study, which is an Alzheimer's disease, a large prospective Alzheimer's disease study, neuroimaging study, they reported that what they saw was a 2-phased longitudinal preclinical AD pattern in which increased cortical thickening was followed by cortical thinning. Their hypothesis was that inflammatory processes were contributing to the increase in cortical thickening, and that was followed by neurodegeneration and atrophy in those people who developed Alzheimer's disease. So keep that in mind. But first, I just want to point out that we can measure lots of endpoints, but really, and lots of endpoints change. But the important thing is, do they have functional relevance for the individual? You know, does it affect how that person or that primate functions? So we asked whether diet composition, these changes in brain, also affected behavior, and we focused on social isolation. And the reason for that is because elders are at increased risk for social isolation, and because social isolation is a really big risk factor for morbidity and mortality. So it's not a small effect, it's a big effect right up there with cigarette smoking. So Dr. Johnson in my lab took all the social behavior observations that we had done, and he did a principal component analysis. And one of the principal components that fell out was associated with changes in behavior, which he called diet-altered behavior. So diet-altered behavior, or DAB scores, are on the vertical axis here. And what you see at baseline is that behavior was identical in the Mediterranean diet to the Mediterranean diet to the Western diet. But by 3 months, behavior had already diverged and remained different in the Mediterranean diet from the Western diet throughout the rest of the study. So
2 minutes remaining.
Okay, so what behaviors are altered by diet composition? Primarily, the Mediterranean diet had spent less time alone compared to the Western diet, and exhibited less anxious behavior compared to the Western diet. So social isolation increases with age and increases mortality in people. The Mediterranean diet increased social contact, also attention and relaxation. The Western diet increased social isolation and anxiety. So at this point, Dr. Negre took a little bit of cortex shown here, lateral temporal cortex, and did RNA-seq, looking at differentially expressed genes, and found that 7 genes in the lateral temporal cortex were differentially expressed. CDK14 is a pro-inflammatory regulator. It was higher in the Western than the Mediterranean diet. These 6 genes are associated with anti-inflammatory pathways. All of them were, transcripts were higher in the Mediterranean compared to the Western diet group. So there's a lot of graphs here, but let me point out that the temporal cortex transcripts we just looked at, CDK14 is in the top row, and the anti-inflammatory ones are in the next 3 rows. And we examined their relationship with circulating monocytes, so peripheral monocyte transcripts, and found that they were associated with measures of inflammation. CDK14 was positively associated with inflammation represented by the CTRA, which is the conserved transcriptional response to adversity, NF-kappa B1, NF-kappa B2, and IL-6. And these other transcripts had negative associations. So we then compared the temporal cortex transcripts with brain volumes. So again, this is a lot of graphs, but if you focus in on CDK14, that's the pro-inflammatory transcript. And the first column is total brain volume, then total gray volume, then cortical gray volume, and the meta ROI, which is gray matter primarily. And you can see positive and significant associations with all of those. So that supports the ADNI hypothesis that the increase in gray matter that occurs early on is in fact due to neuroinflammation. We also looked at the transcript relationships with behavior and found that CDK14 was associated with was positively associated with rates of anxiety and with percent time alone, whereas this anti-inflammatory transcript was inversely associated with time spent alone. So this is just a summary of all of that. So the Mediterranean versus Western diet has impacts on peripheral inflammation, neuroinflammation. Those are accompanied by volumetric changes in the brain, as well as changes in behavior and changes in stress reactivity. Importantly, these afferent signals influence peripheral inflammation, the production of cytokines. So that means that this system can become a feed-forward positive loop. So my conclusions from all of this are that diet's a modifiable risk factor. Poor diet might account for some of the deleterious health effects of low social status. Mediterranean diet may be an efficacious multi-system intervention for aging. A couple of thoughts on translation. Translational value may be increased if lab animals consume diets like those humans eat. Regular commercial chow diets are like nothing humans eat, and they're not like anything that animals eat either. Another thought on translation, long-term studies, the measurements we made at 3 and 6 months were not the same outcomes as the same measurements that we made at the end of this 3-year study. Long-term studies may have different conclusions than short-term studies. The biggest gap I see is that we in the US haven't really done a large long-term diet composition intervention trial. And I'm thinking, for example, like Predimed in Spain that has multi-system aging endpoints. I think that would be extremely useful. My response to many of the earlier presentations was over and over, I heard sex differences in response to diet. And I don't think we know nearly enough about that in human beings. I think in terms of challenge and opportunity, that we need to change the national diet. I think that part is very clear from everything I've heard from you folks and from our findings also. And we can do that. We've done it before, and I point to the National Cholesterol Education Program as one example, and the incredible drop in trans fat content in our food chain after the FDA got busy. So I will thank all of the folks that were involved, particularly Brett Fry, Jacob Nick-Ray, Tom Register, Corbin Johnson, the Wake Forest Diet Lab, our funding from NIA, and the Alzheimer's Disease Research Center and the Claude D. Pepper Older Americans Independence Center. And of course, thank you to the monkeys. And I can take questions if there's time.
Please write your questions in the Q&A if you have any. We could probably have time for one question. Yes, we have a question from a panelist.
Hi, this was fantastic, Carol. One question, you presented some data on diet and stress response. Is there any interaction with socioeconomic status in that paradigm?
I didn't quite hear that. Is there any interaction in?
With the stress response to diet in relation to socioeconomic status, or the dominant versus subordinate monkeys?
Yes, but we didn't see it in the outcomes that I showed you. Those were all main effects. Well, that's not entirely true. There were diet by social status effects in the HPA axis and in the autonomic nervous system. I didn't show them because they're complicated and I didn't have enough time. I will also say that the Mediterranean diet and Western diet have huge impacts on the HPG axis. I also did not have time to talk about that. The Western diet animals have poor ovarian function compared to the Mediterranean counterparts, and that's huge for women because midlife ovarian function really drives late life health in women.
Thank you.
We have one question in the chat, but I think we'll answer it during the panel discussion so we can keep moving along. Thank you, Dr. Shively. That was great.
Thank you.
Next, I'd like to introduce Dr. Callie Thomas. Dr. Thomas is the Associate Director of Health Services Research in the Center for Equity and Aging at Johns Hopkins University School of Nursing. As a gerontologist, Dr. Thomas' research focuses on identifying ways to improve the quality of life of older adults needing long-term services and supports through applied health services research. Her research spans the LTSS continuum, ranging from in-home services to long-term care provided in institutional settings. Today, she'll be presenting on social determinants of good nutrition, understanding food insecurity among older adults.
Right. Thank you for that introduction. So nice to be here today. I really enjoyed the panel discussion so far and the presentations. Quite different from the work I do every day. So I'm here to talk about some work we did to understand food insecurity among older adults. And I think that our approach to this work and some of our findings are really important when we're thinking about the translation and application of precision nutrition approaches among this population. So to get us started, some background on food insecurity among older adults. The USDA defines food insecurity as a limited or uncertain availability of nutritionally adequate and safe foods. And among older adults, it's a pervasive and prevalent problem. It's more than doubled in this population in the last 20 years. And in 2021, it was estimated that 5.5 million older adults, or 7.1% of the older adult population, were food insecure. And the reason that understanding food insecurity and ways to ameliorate it is important is because it's been linked to poor health outcomes. There's research documenting poor diabetes management, worse depressive symptoms, poor blood pressure control among those who are food insecure. Also inefficient healthcare utilization, such as postponing necessarily medical care. We've seen increased rates of emergency department visits and hospitalizations. People who experience food insecurity report having to make trade-offs between healthcare and pay and food. They also are more reliant on poor quality foods, have reduced medication adherence, and food insecurity differentially impacts older adults in specific subpopulations, such as those with functional impairment, multiple comorbidities, socially isolated, homebound, live alone, or in certain parts of the country are more likely to experience food insecurity. And currently the gold standard for measuring food insecurity comes from the USDA, but it only focuses on financial barriers to food, which when you're thinking about an older adult population, financial barriers may not be the only factors that influence their ability to obtain food or nutrition assistance or to adhere to a precision nutrition plan, right? So to expand the concept of food insecurity among older adults, prior research has identified items that can be used to supplement the USDA's checklist or survey questions, if you will, based on the needs of older adults and their ability to access food and nutrition assistance, including their physical abilities, their ability to consume food, as well as their access and use of transportation and social support. And so guiding this model and the way we think about how to identify factors associated with food insecurity or good nutrition, we employ the social ecological model pictured here. And we posit that food insecurity is influenced by these intrapersonal factors, so things like frailty or high levels of medical comorbidity and being homebound, as well as these interpersonal factors that include living alone and a lack of social support, institutional factors, such as the ability to get to the grocery store, the time they have available to cook meals, whether or not they have private health insurance, access to routine health care, community factors, such as do they receive congregate meal services? Are they receiving home delivered meal services? What's their participation in other community benefits and programs, as well as policy and social structures, such as receiving nutritional assistance, enrolled in Medicaid, receiving income assistance. And we believe that all these things are specific to the multidimensional experience of aging. So just to reinforce, I argue that this model can provide a framework, not only for understanding food insecurity, but also how precision nutrition approaches can be influenced and must be informed by these various levels of factors. So in a study that I'm going to present here, we use this model to try and understand which of these different levels and factors are determinants of food insecurity. So we sought to create a summary indicator in the National Health and Aging Trend Study. So more specifically, we identified the prevalence of food insecurity among older Americans. We examined differences in demographic and biopsychosocial characteristics. And then we examined the association of food insecurity with these relevant biopsychosocial factors that have been shown to be related to food insecurity in the literature. So for those of you who are less familiar with the National Health and Aging Trend Study, let me tell you a little bit about it because it is such a valuable resource that is supported by the National Institute on Aging through a cooperative agreement with Johns Hopkins University. It's a national survey that began in 2000, and it gathers information from a nationally representative sample of Medicare beneficiaries who are 65 years and older. The study conducts annual in-person interviews, and they collect very detailed information on factors associated with the disablement process and its consequences. And so in addition to this, long battery of questions that assess all these various domains, they also include vision and hearing assessments. They do CogState cognitive activities. They have accelerometer data, and they have dried blood spot-based biomarkers. So it's like a treasure trove of data for examining any number of aging-related questions. They also do an interview with caregivers in the last month of life to understand individuals' experiences in the last month of life. The other thing I didn't say is this is like a longitudinal, so it can track the same person now up to, 15 years. And then there's a supplement to the NHATS that surveys caregivers, and they're interviewed in the National Study of Caregiving. And on top of all this, all these data are linked to Medicare, like CMS data, so you can identify claims and utilization for all of these participants. So really great data set. So for this particular study I wanted to share with you, we used data from round 5, which was a refresh of the National Health and Aging Trend Study. We included 7,070 community dwelling participants, meaning they weren't in a nursing home or assisted living facility. And then we applied the survey weight. So we can extrapolate our findings from a sample of 7,000 people to 38.8 million older adults here in the US. And we, as I mentioned, because well, I didn't mention this, but the NHATS, despite all its amazing qualities, doesn't have a great measure of food insecurity. So they don't use the gold standard from the USDA. Rather, they have rather we had to create, this is the whole purpose of this, was we wanted to create a measure to understand food insecurity using these data. So in this, we created this new summary indicator that's depicted here using items from the NHAT. So in the last month, did you have to skip meals because there wasn't enough food or money to buy food? How many days did you skip meals? Did you ever go without groceries because there was no one to help shop or you couldn't shop by yourself? Did you ever go without a hot meal because it was too difficult to make it, or there was no one there to help you? Or did you go without eating because there was no one there? So this, as you can see, is a rather severe measure of food insecurity in terms of missing and skipping meals. And food insecurity should be strongly associated with biopsychosocial factors that have been previously correlated with food insecurity. So we examined the association of our new measure with these indicators listed here, specifically a measure of community disability, a measure of social isolation, a measure of frailty, and a measure of homeboundedness. And our results, we included 7,000 community dwelling older adults in our analytic sample. Excuse me, go back. The weighted percent of those that were classified as being food insecure with our measure was 4.3%. And so that extrapolates to about 1.7 million community dwelling older adults. So not just having difficulty paying for food, but like going without food because of financial or functional limitations. Here we present the sample characteristics of those who stratified by whether or not individuals were food insecure according to our measure. And we found that food insecure older adults were more likely to be female, to be Black, to be Hispanic, or in other race. They were less likely to be married, twice as likely to be enrolled in Medicaid. They have more chronic conditions, including depression. They were more likely to live alone, more likely to participate in an aggregate meal program, and to receive SNAP benefits. And similarly, we found that those who were food insecure were more likely to experience frailty, to be semi or totally homebound, to be living with disability, and to experience social isolation. And when we adjusted for demographic and health characteristics, we found that individuals who were frail had 9 times the odds of being food insecure compared to those who were not. In addition, we found that being homebound conferred excess risk of food insecurity, with those who were semi or completely homebound having almost 5 and 3.5 times the odds of experiencing food insecurity. And finally, we found those who were living in the community with a disability had 5 times the odds of experiencing food insecurity. And again, all these models were adjusted for the factors listed here. So in terms of highlighting some strengths and limitations, this was the first effort to create a food insecurity measure in the National Health and Aging Trend Study, which, as you can probably guess from the amazing data and resources that are available in this particular NIH-sponsored study, there have been thousands of research papers, hundreds of grants, and all supported by these data. So we were excited to have a measure that we can now use to understand a pretty severe form of food insecurity among this population. This is a valid nationally representative survey of older adults. It incorporates really important factors that we saw are related to food insecurity, including frailty, social isolation, community disability, and being homebound. And it also demonstrates unmet need, that some food insecure older adults did not receive meal assistance, specifically that finding around SNAP. I'll go back to that because we would have hoped, I think it really calls for a need that only 16% of people who experience this level of food insecurity were receiving some kind of federal and state supplement for their nutrition. Okay. So in terms of limitations, as I've referenced a few times, this measure really captures a more severe form of food insecurity, which is probably why the rates we see reported here are lower than rates we've seen in other samples or even in the national population using the USDA measure. We also, the other limitations is it doesn't include people who need help, but have help. Because the questions, as you saw in our measure, were among those who either couldn't do it themselves, like didn't have help, or didn't have someone to help them prepare, obtain meals and groceries. Also, because the NHATS relies on Medicare enrolled beneficiaries, it excludes individuals who are homeless or who might not be Medicare enrolled, either because they don't have enough quarters or they aren't citizens but are still here in the country. As well, the items in the NHATS aren't as comprehensive in measuring food insecurity, such as the USDA's HFSSM measure. And finally, given the sample size, we were unable to explore differences among racial and ethnic minority groups, which I think is really important moving forward as we think about different factors that influence food insecurity and how those might vary by demographic groups. So in conclusion, using data from the NHATS, we created a summary indicator of food insecurity among community dwelling older adults. This measure is found to be correlated with biopsychosocial limitations that older adults disproportionately face. And in terms of future directions and future work, I think it's really important that those that target food insecurity, as well as just good nutrition broadly, must consider these biopsychosocial factors and how any efforts that we promote either address or figure out a way to to navigate those in our work. And finally, precision nutrition efforts must consider the needs, resources, and limitations that many older adults have, including the same limitations and the same factors that influence food insecurity. I want to acknowledge my co-authors on the work I presented here, Dr. Emma Tucker, Dr. Tamara Keeney, Dr. Alicia Cohen, funding from the National Institute on Aging, and I just wanted to disclose that I have nothing to disclose. And then last, I wanted to share, if you're interested in learning more, this is the publication that details our work that I presented today in the Gerontologist. And also wanted to share that we have an ongoing study from the Patient-Centered Outcomes Research Institute that focuses on understanding 2 different modes of meal delivery to homebound older adults. And the reason I highlight this work is that because we're also learning about and investigating the influence of many of the factors I referenced today and how it relates to health outcomes and healthcare utilization. So I'd love to share more about this trial that's currently ongoing and some of our early observations, as well as answer any discussions or any questions during the discussion with my fellow panelists. Thank you all very much. If you have any questions or comments that we can't get to today, my contact information is here. Thanks.
Thank you so much, Dr. Thomas. That was excellent. And we do have time for questions. Please, if you have any, write them in the Q&A section. You touched on it a little bit. I have one. So what does a food insecurity intervention look like? Or I mean, you were kind of touching it in your PCORI study, but I was just curious, considering everything that you've presented, it seems very multifaceted, and I'm just,
Yeah, no, I think it absolutely is. And it's going, and any kind of strategy has to be thoughtful about the population it's targeting and its implementation of that. So I think the reason why we still see such high rates of food insecurity is because we don't have a great solution at the moment, right? With SNAP, SNAP has been shown to reduce food insecurity. It's a really great program. However, as you saw in this data, particularly among the older adult population, it's underutilized. And so I think even understanding barriers to enrolling in SNAP is very important for this population, because that is sort of one lever we have to address food insecurity, particularly the financial hardships of food insecurity, or that influence food insecurity. Yeah. I'd also like to argue that I think increased investment in senior nutrition programs like Meals on Wheels that bring food, as well as, so they provide congregate meals for older adults to come and socialize and receive meals, but also importantly, deliver meals to homebound older adults who are unable to either obtain their own food or stand in front of the stove to cook their own food. And unfortunately, the budget devoted to those programs is like rounding error on many state and federal budgets. So I do think they're great programs that our research has shown influences health and health outcomes and food insecurity, and that we're working to continue to build the evidence base to get more support for that work, or for those programs.
Great. We do have a question in the chat. Hi, Callie. Nice presentation. Quick question. Is there a plan to study needs for racial ethnic group F1 trends in future? Several community delivered meals may not cater to culture specific needs.
Oh, great question. Hi, Brahma. Yes. So, this is the cultural tailoring of meals is something that is very important to older adults. And it's something that I think the aging network and senior nutrition programs struggle with more broadly, particularly as we see increasing diversity in the older adult population here in our country. So one of the things we're looking at in our current work is around how these different modes of meal delivery, right? So meals, specifically thinking about home delivered meals. If meals are delivered by a volunteer or paid driver from a program that has a caterer and has a menu of meals that a person gets, think traditional Meals on Wheels every day, versus these frozen meals that provide more flexibility to be tailored to people's preferences and desires, if those differentially impact satisfaction, experience, adherence, health. So we're doing some work to dig into that, but I think that much more needs to be done, as that's such an important part of, adhering to a good nutrition plan, or nutrition in general, is having meals that people enjoy and want to eat.
All right, we have a question from you.
Hi, great presentation. So I have a quick question. So I may have missed it. So you've listed you've listed factors that sort of lead to food insecurity. So do you also have a sense of in terms of health outcomes, what type of health outcomes this population are most vulnerable to?
So, there's been a pretty, I would say, large body. It could be larger. We need more work in this area that's been done to look at how food insecurity impacts health outcomes. A lot of it is like association based versus, I would say there's not many studies that either use an experiment or causal inference, but those that do have shown that. I think, on the interest slide that I shared, are related to poor, like diabetes self-management, for example, increased risk of healthcare utilization. But I think more work to understand like how much, which direction is that going, would be really important. Does that answer your question?
Yes.
Yeah, okay.
Yes.
Right.
All right, well, thank you again, Dr. Thomas. And we will go ahead and transition into our last speaker of the session before the panelist group is Dr. Sean Gibbons. Dr. Gibbons is Associate Professor at the Institute for Systems Biology in Seattle. He investigates how the structure and composition of evolving ecological networks of microorganisms change across environmental gradients. In particular, he is interested in how ecological communities in the gut change and adapt to individual people over their lifespans, i.e. host genotype, host development, and host behavior, and how these changes impact human health. Specifically today, he'll be speaking on designing microbiome-targeted interventions that promote healthy aging. So with that, the floor is yours, Dr. Gibbons.
Thanks, Andrew. Do I have the wrong screen up, or is that the right screen?
No, you're good to go.
I'm good to go? Okay, great. Thanks for that introduction. Thanks for the invitation to this series. The other talks have been amazing. I've been learning so much. Now we're going to talk about the microbes, which I haven't come up that much as of yet. So yeah, my group studies the human microbiome and how it is a mediator in how we respond to various exposures, diet, drugs, so on and so forth. And just to give you a quick background, if you're not familiar, we're about as microbial as we are human by the numbers, as about 30 trillion human cells in the body and something like 40 trillion microbes, although depending on your last bowel movement, you might be more human than microbe. Even though they're much smaller than our cells, so they're only about half a pound of material. But then if you look at the genetic capacity of these organisms, they really swamp us. So there's 23,000 or so genes in the human genome and something like 2 to 4 million unique genes in our microbiome. And a lot of that genetic capacity we leverage for our own phenotype, right? We've outsourced some of the functionality of our bodies to these organisms, including digestion of complex plant polysaccharides, training of our immune system, and so on and so forth. We know that our genomes give us these unique responses to diet or other kind of inputs throughout our lives. But the microbiome, as we move along, we're learning more and more. It's another one of these filters on our phenotype and can kind of determine what direction we go. Even if you have an identical twin where you share the same genome, your microbiomes are about as distinct as two strangers. So even twins have very, very distinct microbiotic composition. So that's quick intro to the microbiome. And I'll jump right into one of our papers from a few years ago. This is really our first foray into the aging space. And this is work in collaboration with two of my colleagues, Eric Orwall and Nathan Price, who got us access to some human cohort data. And this work was really driven by Tomasz Womansky and Noah Rappaport from Nathan's group, and Christian Diener and Sushmita Patwardhan from my own group. When we started this project, we were looking at the literature, the microbiome aging literature, and there were kind of two different signatures that were apparent to us. One was in people kind of more frail and assisted living facilities where we see kind of a maintenance of the core microbes throughout the lifespan that are in the adult gut. And then the other was in centenarians, where you actually see a steady decline in the dominance of these core taxa that tend to dominate us when we're younger, and they're changing and getting less dominant with age. And these subdominant organisms that actually make us all more distinct, these are rising in dominance with age, but only in these healthier people or in these centenarians. So there seem to be kind of signatures of healthy aging and then other kind of signatures of aging. So we want to try to tease all of that apart. And in order to do that, we had two large data sets that we pulled down. One was called the Aravel cohort, and this is it was a consumer company, essentially. A bunch of people paid a premium to have their bodies quantified, soup to nuts, microbiome, your blood metabolome, clinical chemistries, and so on and so forth, Fitbit tracking. And so this cohort spans the human adult lifespan almost, right? It's between about 18 and 90 years old. About the midpoint is about 48 years old is the average. About 3,600 people with a full suite of data, mostly a female cohort, about 60% female, 40% male. And generally speaking, these people are biased towards being a little healthier than the average American. It's mostly people on the West Coast of the US, in Washington State and California. Then we had an older cohort. This was called the Mr. Ross cohort. It's a cohort of older men that was originally recruited to study osteoporosis in men. It's about 1,000 men between around 80 and 100 years old, essentially. And for this cohort, we also had microbiome data and paired data on health and so on. And we had natural splits within these cohorts. We could have sort of training and validation sets across all of them. We also pulled down the American gut cohort, which doesn't have host phenotypic kind of molecular measures, but it does have microbiome and age data. So just jumping right to the main point, we wanted to see if we could see the centenarian-like signature in the data. What the centenarian papers have reported is a decline in core taxa, but if you go to different parts of the world, there's different core taxa, right? If you're in India, you're probably dominated by Prevotella in your gut. Whereas if you're in the US or Europe, you're probably dominated by the genus Bacteroides, and these things are kind of mutually exclusive. So rather than picking a core taxon that's consistent, we kind of took the inverse of that, which is to say, what makes you more unique? So if you have a less of these core taxa that make us all more similar, you should actually look more distinct from your neighbors if you have all these subdominant taxa rising in prevalence. So we came up with this uniqueness score, just how close are you to your nearest neighbor in the data set. So this score requires having kind of a larger cohort of people to compare others against. And generally what we saw was this steady drift as you get older and older towards the later decades of life, people's uniqueness rises and rises. So we're all kind of drifting apart from one another as we get older. And again, this is a largely kind of healthy population of people. It starts in your like 40s or 50s, and it starts to kind of accelerate more and more as you get older and older. And this is true across all of our kind of different test and validation sets. Even in the American Gut cohort, you can pull that down, you see this as well. There's a few dozen papers now that have kind of seen similar patterns in other cohorts, human cohorts, and even in mice now at this point, I think. So, what are the taxonomic signatures associated with this uniqueness drift? This is a high level plot just to give you a basic flavor for the main patterns, which is a negative association with gram-negative anaerobes like Prevotella and Bacteroides. So high uniqueness means lower levels of these core genera. And there's a positive association with a pretty wide diversity of different genera within the order Clostridiales. So kind of butyrate producing or short chain fatty acid producing gram-positive anaerobes are positively associated with this uniqueness score. But everybody has kind of a different set of them, and so that's what's driving this drift or uniqueness signal. Now, despite this drift, ecologically, people are becoming more and more dissimilar ecologically. If you take this uniqueness score and associate it with what's happening in the blood metabolome in this same Aravel cohort, there are certain things that are consistently associated with this uniqueness drift. And these are the hits that come up. By and large, they're mostly protein fermentation byproducts, so kind of fermentation products of amino acids like tyrosine, phenylalanine, and tryptophan. And they're higher in the bloodstream of people with higher uniqueness scores. And this is, to me, very interesting, and it kind of feeds into some of the prior talks of the day, because some of these aren't necessarily great, right? Some of these are toxins to like the kidneys, right? Like p-creosyl sulfate is a risk factor for disease progression and chronic kidney disease. So not all these are, and indoxyl sulfate is another one of them. Not all of them are great. Phenylacetylglutamine is also kind of a iffy on the fence, whether it's good or bad metabolite. But if you go and Google it, it's actually patented as a biomarker for centenarian status. So not only is it like super elevated in the blood of centenarians, but it's elevated in their children's bloodstream as well. So it might be either a genetic or a lifestyle marker for the chances of achieving a longer life. Some of these look like they could be beneficial as well. So indoles, for example, have been shown to reduce inflammation and even extend health span in mouse models. And then some of these secondary bile acids similarly have been shown to extend health span and lifespan in Drosophila and in mouse models. So yeah, we definitely see this uniqueness manifested in the bloodstream, kind of with an ambiguous signal there. And then looking kind of at the beta diversity, where the distance between individual samples and in community structure space, so projecting the multidimensional microbiome composition down into 2 dimensions, you can clearly see that having higher uniqueness puts you in the elbow of this distribution. High Bacteroides is on this arm, and then high Prevotella is down on this arm. And this elbow is also enriched for these gram-positive anaerobes, essentially. And so there's a very strong, consistent negative association between this uniqueness and the dominance of these gram-negative anaerobes. So this is all true in the Aravel cohort, but in the Mr. Oz cohort, our signal got weaker. So this association between aging and microbiome uniqueness actually was dampened. It was still significant, but it was dampened. So because we had health metrics for this cohort, we wanted to separate people out and see if we saw variable signatures going on. And that is what we saw. So by a number of different metrics, walking speed was one, self-reported health, number of medications used, and the life space score, which is a sort of score of motility or how much you get around in your life. By any of these metrics, if you report being healthier, we saw a nice significant association between uniqueness and increasing age. But in people who reported being less than healthy, that association was ablated. We no longer saw it. And it was true across our sort of test and validation cohorts. And we had a composite score as well, where you had to be healthy according to at least 3 of the other scores to be composite healthy. So this nice consistent signal. And this is all observational cohorts, right? There's no time here yet. But in Mr. Oz, we actually did have follow-up data. So we had a 4-year follow-up with baseline data and then a 4-year follow-up. So we could ask, did the composition of your microbiome at baseline tell you something about your odds of survival in the next 4 years? And it did. And so whether you looked at this uniqueness score or whether you looked at the dominance of the main core genus Bacteroides in the population, if you had a lot of Bacteroides, you were more likely to perish in the next 4 years. And if you had low uniqueness, you were more likely to die. And with Cox proportional hazards regression, you could control for some of the potential confounders for mortality, like markers of diabetes and cardiovascular risk. And so even when controlling for all of these things in this last column here, we still see significant association between this microbiome signature and the likelihood of survival at follow-up. We're still not sure what the exact mechanistic interpretations are for what these signatures are and how they might be just correlated with aging or and healthy behaviors, or if there's some sort of causal thing going on with the microbes. We started to dig into metagenomics to kind of look at gene abundances and whether those are associated with uniqueness and aging. And we do see some stuff that makes sense. So, for example, we find that generally speaking, in people who show this healthy looking signature in their guts, they show a decline in the abundance of genes involved in degrading host glycans, like your mucus layer, for example. A lot of the species in the Bacteroides genus are facultative mucus degraders, and they will eat you, they'll eat your mucus, especially if you're not eating a lot of dietary polysaccharides. And so perhaps a decline in these organisms with age, because in the aging gut, the host actually stops producing as much mucus. And so the mucus production rate declines. Thinning mucus layers leads to inflammation, and that's generally bad and thought to promote aging. We also see genes involved in dampening inflammation responses and immune responses also being associated with this uniqueness score. So we're not exactly sure what's going on. I think a lot more work needs to go into this, but at least we can find these molecular associations and observational cohorts, and then perhaps take those data back to a model system, a preclinical model, and try to understand mechanistically what's going on. So I'll quickly brush on some prelim data we have with a collaborator on this very idea of reverse translation, right? You observe something in humans that might be microbiome mediated, and then you try to test that in an animal model. Here we're looking at NMN supplementation. NAD is widely reported to kind of decline with age. But it's unclear whether supplementation with NAD or its precursors like NMN really does have a material effect on lifespan or health span in humans, or even in animal models. And so Alice Kane, who's my colleague here at ISB, did a long-term supplementation trial in mice for about half their lifespan. She's supplementing them with this NMN supplementation and looking at phenotypes. So she did see an increase in health span, but I don't think lifespan in female, but maybe not male mice. So there were all these kind of sex-dependent effects. I won't talk about the lifespan, health span stuff. I'll talk about what we looked at, which was associations with what's going on in the gut. So we found that generally when you supplement with NMN, you don't get a huge effect broadly on the community structure. There is a significant effect between treated and untreated, but it's sort of subtle essentially in both males and females. But if you look at specific differential abundance of taxa, there was one organism that emerged as like screamingly significant, and it was this bug, Anerotrunchus collihominus. And this is one of these Clostridioides taxa. It's a butyrate producer, essentially short-chain fatty acid producer, and it is greatly increased in its abundance upon NMN supplementation, and that is maintained throughout the rest of the life of these mice, both for males and females. And Alice was collecting frailty scores for these mice, and we found that controlling for age as a covariate, we saw a significant association between the abundance of this bug in the guts of these mice and frailty in the females, but not for the males. The males was sort of a trend, but didn't approach significance. So that's talking about our work on aging, the kind of the full sum of what we were doing on aging so far. We're kind of moving more into this area, but I thought then I'd pivot quickly to some of our other work on precision nutrition and how it could be relevant to the aging context, but we haven't quite applied it there yet. I don't have a lot of time to give background, but really talented research scientist in my group, Christian Diener, who now runs his own lab at the Medical University of Graz in Austria, designed this tool called MyCom, which is we're extending flux balance analysis, metabolic modeling of individual organisms to complex multi-species communities like those found in the gut. You initialize the models by having constraints on the composition of the microbiota. So you sequence someone's gut, you see who's there, what abundances, and that can be a constraint on sort of a digital twin, if you will, of someone's microbiome. You can constrain the dietary input, whether they're eating a Mediterranean diet or a standard Western diet. You could even have personalized dietary constraints, although those are kind of hard to build, to kind of convert, like a carrot into the actual molecular constituents of a carrot, but it's possible. And then you get this, prediction of what is being produced by the microbiome, what metabolites are coming out, what it's consuming from the diet, the interaction, the cross-feeding, and all that's going on in the system. And so it's been widely used and cited the last few years. We're happy that people are using it, and we're finding it to be potentially translationally useful. So that is what I'm going to get into next. There's a key challenge to translating the microbiome into the healthcare context. A lot of what we do so far is like fecal transplants. They're kind of like a hammer. We're just doing a whole ecosystem transplant, and we don't always understand the mechanisms at play for how that's driving the phenotype. But in order to get precision, towards precision, we really have to understand exactly mechanistically what's going on. And one clear problem is if you feed a bunch of people a banana, you get completely different short chain fatty acid profiles out the other side. And we don't really quite yet know how to predict that, except perhaps now we might. So we've gotten some traction on that recently. This is work driven by Nick Quinn Bowman, a really talented PhD student in the lab who just defended. He's now Dr. Nick, along with Catherine and Crystal, who are running the wet lab experiments. Christian again, and then our collaborators at the Fred Hutch, Joanna Lampe and Lisa Levy, who are recruiting our patients who are giving us really, really fresh stool. We get stool right out of the chute within 30 minutes into our anaerobic chamber for running our experiments. So here the question is, if you feed a bunch of people inulin, which is the fiber in a banana, can we predict that inter-individual variability in butyrate or propionate production? And just to focus in on butyrate a little bit, if you're not familiar with it, there was some talk about ketones earlier. This is very similar to beta hydroxybutyrate. You take away the hydroxyl group. These are short chain. This is a short chain fatty acid. It's a particularly potent one. It has very vast array of effects across the body, both in metabolism and in signaling, like was discussed for ketones. So, for example, it increases insulin or your ability to control glucose in the body. It reduces insulin resistance. It helps improve bone density. It helps with weight loss. It helps stimulate GLP-1 production and PYY production. With that, helps with appetite. Cardiovascular risk is reduced with more butyrate production. It's kind of an all-around healthful molecule that is produced exclusively by our microbiota in the colon from fermenting fibers, essentially. But people produce very different levels of butyrate depending on who they are and what their microbiome composition is. So how do we test this inter-individual variability? Is that my 2 minutes?
Yes.
Okay, thank you. We actually have to take poop out of the body because in vivo, these SCFAs are rapidly absorbed and degraded by the host epithelium. 95% of all butyrate is consumed by our colonocytes as the preferred energy source, even more so than blood glucose. So that it's gone as soon as it's made. So in order to measure its production, we have to take it out and measure these fluxes in time series metabolomics assays, looking at stool in vitro cultured anaerobically. So we do this, we can predict, we have some validation data now showing we can predict butyrate and propionate with some level of quantitative accuracy, not perfect. But this isn't a statistical model. This is a mechanistic model, and these predictions are emergent properties from the optimizations of biomass in the metabolic model. So we're pretty happy that we're getting any level of prediction here. We can see that these predictions are related to clinical chemistries in this Arabelle cohort. So if you're predicted to produce a lot of butyrate, you see lower levels of LDL cholesterol, triglycerides, so on. You see better control of glucose. You see lower inflammation. And if you measure blood butyrate, you see no association with any of these things. So blood butyrate is like white noise because I think it's very homeostatically controlled and not very much produced butyrate in the gut is actually making its way into the blood. We see that, in a trial to bring down blood pressure with a resistant starch, we can explain the responders and non-responders in that trial based on our butyrate predictions. We're trying to implement these precision interventions. So someone doesn't respond to a high fiber diet, can we predict co-supplementations that can rescue the non-responders? So that's what this cartoon's showing. We add either a prebiotic fiber or a probiotic butyrate producer, Faecalibacterium, to the background of a high fiber diet to try to take people who are non-responders to butyrate production on a high fiber diet and turn them into responders. So that's what these distributions are showing. The dark blue is the precision intervention, and those precision interventions are now plotted here, where every column is a person. The black box outlines the optimal intervention for that individual person via our simulations. And you can see that for some people, adding a simple dietary fiber to the background diet really turns up butyrate production. But for others, you require a probiotic intervention. If you lack the metabolic capacity to produce a lot of butyrate, you're not going to make it no matter how much feedstock you push down the system. So I'll stop there. I'll just say, I think we need to think better about defining healthy aging from the sort of diseases associated with aging and the negative consequences of aging. Not all changes that are going on are necessarily bad. Some stuff is compensatory. I would argue you maybe don't want to push the centenarian microbiome back to a 20-year-old's microbiome. That could lead to maybe mucus layer thinning and other issues. So don't take FMTs from young people into old people potentially. And I think these precision approaches are really going to be helpful to design interventions that help people more broadly. Thanks to those who did the work, and thanks to all of you for listening.
Wonderful talk, Dr. Gibbons. We have a couple questions in the Q&A that we can take real fast. First one, fascinating work. Have you looked into cardiac health profiles in your data sets and wondering if Acinetobacter may have popped out anywhere in your studies? Acinetobacter. Yeah.
Acinetobacter pops up here and there. Yeah. I can't, not in any of the things I presented today, I don't believe so. But we do look at cardiovascular markers. I think I showed in the clinical chemistries, we saw lots of these lipid markers of cardiovascular risk were negatively associated with butyrate reactions. And in this HAMSAB trial, we see that you can bring down blood pressure with resistant starch. The trial showed this, but a large fraction of the population is non-responding to that intervention, and we can kind of predict that a priori, it seems, with our model and show which people are going to be producing more or less butyrate on that particular intervention.
And that kind of leads into our next one, and you touched on this a little bit in your talk, I believe, but can butyrate be supplemented? And what are your thoughts on oral butyrate supplements?
Yes, it can be. And in fact, that HAMSAB trial, the AB on the end, the HAMS is, I think, high amylose maize starch, and the AB is a conjugated acetate and butyrate. The problem with giving pure butyrate to someone in like a pill or even as a gavage or something is it's really stinky. You'll smell like a sewer if you try to do that, and it's really not tolerated well. They used to give like butyrate enemas to people with inflammatory bowel disease and stuff like that. It worked to bring down inflammation. It was therapeutically effective, but I think patients didn't really tolerate it because it was so stinky. Some of the conjugates, like conjugating it to the starch, it cuts the stink a little bit, but from what I hear in that trial, it was still pretty gross to eat that starch. So that's the main issue. It's also difficult to deliver the amount of butyrate that is really required in to be produced at the periphery or like at the epithelium. And so I think it's probably most efficient, generally speaking, to allow your microbes to be continually excreting these SCFAs by giving them the dietary substrates that allow them to do that.
Okay, great. A couple more questions just rolling in. Do people who live and age together have more similar microbiomes than those who do not live together?
They do, yes. There's great work on this. Nicola Sagata has some interesting strain sharing work on mothers and infants and fathers and infants and people who are romantic partners. What you generally see is that adults who have an intimate relationship with one another, they can share like 1% to 2% of the strains that reside in either of their guts. So it's not a lot. It's like a very small percentage. But if you compared two strangers, they would share 0% of strains, right? At the strain level, they would have no sharing. So you get very small levels of sharing. The highest levels of sharing you get between individual humans is when you look at young siblings. So little kids that are between, 1 and 3 years old, they can share, up to, 30, 40% of their strains sometimes. Because I think the microbiome at that point in time is a little more plastic, and they're maybe interacting a little more physically than the average person, spit and all kinds of stuff. So I think that for whatever reason, you get most sharing in siblings, and it drops off from there.
All right, and last question before we move into the panel. How might the host genetic composition influence gut microbiota composition?
Great question. We actually had a paper on this a couple of years ago in Nature Metabolism. So you can look up me and genome microbiome interplay in the blood metabolome. And essentially and there's other papers on this as well, but there's very little association between the host genome and the microbiome. There are some microbes in the gut that have small amounts of their variants explained by host genetics, but it's like in the very small single-digit percentages of their variants explained, and most stuff isn't really. So diet is a much stronger factor shaping composition than genetics. Genetics is almost invisible to the microbiome, it seems. And then when you look at the metabolome, the effects of microbes and genes on circulating metabolites appears to be orthogonal. So whatever the genome is doing is kind of separate and apart from whatever the microbes are doing.
All right, great. We have a couple other questions, but I think we'll invite all of the other panelists back up and then we can discuss together. So if you have any questions for the panelists to address together, please write them in the Q&A. If not, I can revisit some of the older questions that we had that we didn't have time to get to. One that I kind of wanted to get to that was addressed to my presentation that I think matches really nice with Dr. Gibbons' presentation was this question about someone, if they're working at night, being able to adapt their schedule or their melatonin rhythm to working at night. And you actually can't unless you're working on like an oil rig or somewhere where you can have a very tightly controlled light dark cycle and not have to interact with your social or family environment. Because our clocks are very rigid. They don't want to shift, but they are slightly flexible. So if we travel somewhere new to a new time zone, we can shift our clocks very slowly. And that's why we experience jet lag, is because our clocks haven't caught up yet. The problem with shift work is it's such a drastic shift that you can maybe start to, after a couple of days, start to shift your clock. But as soon as you have that weekend of non-shift work, you're going to go right back to where it wants to be. So we think of that as a misalignment, but how it pertains to Dr. Gibbons' talk is that we are thinking about this in terms of an internal desynchrony. And so when we're awake and at night and we're eating during that time, we can start to entrain some of our clocks. So we have circadian clocks throughout the body, but some in the gut. And recent work has suggested maybe in the microbiome, we can shift those clocks to actually meet that time. And then, so you have a desynchrony from your central clock to your gut clocks. And that could be what is resulting into a lot of the poor health outcomes that we see in shift work and after, because we see after effects after someone has shifted back, we still see after effects, and it could be because some of the gut clocks. And so I was just curious, Dr. Gibbons, what your thoughts might be on measuring clocks in the biome, and if you'd be open to collaboration, because I can definitely provide samples. So.
Totally. So yeah, actually, and when I was in graduate school, I was a co-author on a paper that was looking into this very thing, Gene Chang's lab at UChicago. And, unfortunately for humans, we poop about once a day, right? So it's hard to do diurnal cycling measurements in the gut for humans, unless you have like robot pills or something. But for mice, when they're pooping every 5 minutes or so, you can get samples across the day. And so we did an experiment looking at how the high-fat diet versus the normal chow diet. The high-fat diet seems to mirror what happens in shift work. It sort of helps to desynchronize the peripheral and the central clocks in the host. And the mechanism, what we found was going on, at least in these mice, was that in the high-fat diet, the mice would kind of eat, they would snack throughout the day, and they'd eat a little bit here and there. On the normal diet, they would actually eat a big bolus of food, and they would get full, and they would stop eating for a while. And so you'd get this bolus of material that led to a pulse in SCFA production, butyrate in particular. And that pulse seems to help to entrain the peripheral and the central clocks with one another. And you could rescue the mice and their disrupted clocks by just directly pulsing butyrate into their bloodstream as an intervention.
Fascinating. We have a question from Dr. Newgard.
Yeah, I'd like to ask about the food insecurity talk, Dr. Thomas. Thank you for that. I'm working here at Duke with a colleague, a pediatrician named Sarah Armstrong, on pediatric food insecurity, both in clinical and preclinical studies. And as you know, food insecurity in pediatric subjects paradoxically can lead to obesity, right? To obesity and metabolic disease phenotypes. So I'm just wondering, do you expect that what one learns in trying to study pediatric food insecurity, how do you think that might inform the type of food insecurity that you're studying in more elderly subjects, and vice versa?
Wow, great question. I actually did not know that about, I'm not following the children's nutrition work at all. So I haven't thought about that. It's tricky because I find, so I'm a trained gerontologist, and I find we often sort of equate like older adult issues to issues, to childhood issues, both in terms of thinking about autonomy, especially those, that are experiencing cognitive impairment. But, I hadn't thought about how the findings in children and its effects on health would relate to what we're seeing with older adults. Yeah.
One thing I thought about was, if this is brought about by epigenetic or other kinds of reprogramming, right? In the younger subjects, how permanent is that? Or how recurrent can it be and worsened by subsequent rounds of food insecurity that may occur later in life, it was just a thought for people to chew on, stimulated by your talk. Thank you.
Well, thank you. And I'd love to hear other panelists' thoughts and remarks on that too. But yeah, I think it's a great question.
Yeah, I don't know if I have too many thoughts on it, but it reminds me a little bit of the last session talking about how like protein has differential effects in early life or in late life, and how diet effects can really vary across the lifespan. And it sort of meshes with this weird observation we have of like these toxicants that are really elevated in the bloodstreams of centenarians and healthier older people, but maybe they're also associated with a higher protein diet in that elderly population, and maybe that's good for them, but maybe bad for a younger person. So I don't know. I think this is like interesting to think about.
Yeah, I think it really gets at that precision medicine aspect of this whole talk. I think that's why the question was asked mostly.
Yeah, life course perspective is important for sure.
Speaking of that, we have a question in the chat. Could individual age impact butyrate production?
Well, butyrate production is, in the body predominantly is pretty much only done by the microbes. And so in so much as aging is affecting the abundance of these butyrate producing organisms, and the community context can also actually control butyrate production or short-chain fatty acid production. If you have cross-feeding with other organisms, it can actually help you produce more of a particular metabolite. So it's not just the butyrate producers that are important. So yeah, shifts in the community structure that are aging related could be affecting butyrate production. But I don't know if that's true. One thing that I'm curious about, and people here probably know a lot more than I do, changing dietary patterns as people age. What kind of foods do people naturally select? Does appetite decline with increasing age? Because if you have less dietary polysaccharides coming into the system, you just, the feedstock for producing these short chain fatty acids is declining. And I suspect that might be happening with many people in aging, but I'm not sure.
Thank you. Dr. Shively, I was kind of thinking of your take homes of improving diet, Mediterranean diet, to potentially lower SES communities, and then Dr. Thomas's ideas to increase Meals on Wheels and provide, reducing food insecurity. And I was wondering if I'm thinking that there are a lot of connections between the two of ways to improve diet to these communities that need it. And I was just curious kind of your thoughts on that, or kind of if you've thought about how you would start to implement some of these take-home messages that you have.
Yeah, I mean, I think it's very important. If you could only change, one segment of the population in terms of their of the food they eat to affect like the number of healthcare dollars we spend in the country, you would want to focus on low SES. So the low SES folks have lots of barriers, but certainly it's the food desert problem. So, and I think we're all pretty familiar with that. There's just access problems to good food. There are ways to increase access to good food without it being horrendously expensive, but it definitely comes from has to come from a policy down or community out kind of an approach, I think. So I think that would be extremely helpful. I also think that so many of the subsidized foods in this country are not really very good for you. And so the foods that our government subsidizes. So some of them they're cheap because they get subsidized. And so they're consumed a lot and they're very readily available, but they're not necessarily good for our population, for our communities. I think a really close look at the subsidy situation and how it impacts the health of the population would be very helpful. There are almost no subsidies for fruit and vegetables. So, you could start there. Anyway, Callie.
I would love to piggyback on that. So I was part of a pilot. I didn't lead it, so I can't take credit. A few years ago, that happened at Brown University, and they used incentives for SNAP. So people got incentives if they used their SNAP dollars to purchase food and vegetables, and they saw an increase in food and vegetable consumption, improved dietary intake, like all these amazing outcomes. But yeah, getting that sort of that disseminated and taken up has been a bit of a challenge. So I think you're right. You're right on track with like the incentives have to align with healthy eating behaviors, and there's research to suggest that it can. It's just making it happen.
Right, and I mean it has to be affordable, and then folks need to be incentivized to pursue those kinds of foods. One of the best impacts, but only on a tiny scale, are community gardens. But they really work. Right around a community garden, the food intake, the food quality is much better than further away from these community gardens. We got excited and built a community garden in our little town, and a Girl Scout troop. So it was amazing, and all the food that people couldn't consume because they produce so well goes straight to the food pantry. Well, who's at the food pantry? The folks that really need the fruit and vegetables. So I think that there are ways that you can build these networks out, but they're not being done subsidized, funded at a great enough level at the moment.
Yeah, that's great. We did a farm share during COVID, and I've never eaten so much squash.
Yeah, exactly.
There was a question about, to me, about evaluating submarine Navy folks, a captive group eating the same time, no light, and they're underwater for months. There is quite a bit of work. The Navy does quite a bit of work looking at sleep and circadian rhythms, particularly in submarines. Even kind of crazier is until fairly recently, they used to put individuals on a submarine on an 18-hour day. And so every day they would wake up 6 hours earlier. And so they were creating a circadian desynchrony on top of everything else because they don't have enough room for everyone to sleep. So they had to hot bunk and they would rotate it around. And so until fairly recently, they just changed those practices. But they do look at that quite a bit. And then is it possible, kind of connecting the two sessions from today, is this possible that ketogenic diets can result in circadian rhythm disruptions or on the microbiome?
Good question. I'll just say quickly that from the mouse work that I talked about earlier, in the context of those mice, it did seem like having like a big meal of dietary fiber that had led to like a pulse of short-chain fatty acid production during a confined window of the day was helpful in training those clocks and helping with metabolic regulation. So ketogenic diets that might be really low in undigestible fibers wouldn't provide such a pulse. And so maybe that's not great. So maybe you should think about supplementing indigestible fibers into these types of ketogenic interventions.
Yeah, I currently have a randomized clinical trial looking at crossover design, looking at a high-fat diet versus low-fat diet on our ability of our clocks to synchronize, because there's evidence from the animal models that it makes our clock sluggish. And I had that preliminary data looking at across daylight savings time, but we're really trying to really tease out the mechanisms and if it's a reduced sensitivity to light or what's happening there. But we haven't done it with a high protein per se. It's more, low carbohydrate, high fat diet. So. Okay, and then kind of piggybacking on the microbiome, I wonder if those people with banana eating have similar blood chemistry profile or inflammatory signatures, even though they had microbiome variability.
Okay, I'm not sure what the question is.
If they had similar blood chemistry profiles or inflammatory signatures, even though their microbiome was variable.
So for the people that we made these poop soups for, we didn't unfortunately have the blood clinical chemistries. So I can't directly answer that question. But indirectly, for that Arabelle cohort, we did have paired microbiome and blood chemistries. And we find that those people who produce more butyrate, so we made a dumb assumption and we fed everybody the same average European diet. And I think it's technically the Austrian diet, which is a little bit of a meat-heavy diet. If you feed everybody that diet in silico and predict how much butyrate they make, that amount is negatively correlated with inflammation, LDL cholesterol, insulin resistance, and a bunch of other stuff.
Great. And then there was a question about meta-analysis on how Mediterranean diet may impact circadian alignment or misalignment. I am not familiar with any on the Mediterranean diet per se. Dr. Shively, do you have any idea?
No, I don't think that I've seen any data on changes in circadian rhythm with Mediterranean diet. We had the 24-hour ECGs and activity also. They were actimeters. And we didn't see a difference between the Mediterranean and the Western diet. So, and I'm not aware of any human studies.
Great. And I think we have 1 minute left, so I think I'll leave it open if there are any other questions or if any of the panelists. I think I did have a. Oh yeah, what is the impact or role or contribution of oral flora changes in older adults to gut microbiota and health?
Good question. Yeah, like something like 95% of all the biomass in your body is in your gut. So the oral microbiome by biomass is like a very small component of what's happening in your gut. So we are swallowing oral microbes all the time. They're washing through, they're detectable at usually really low levels in stool. The only case in which you see higher levels of detection of oral associated organisms in poop is in cases of colorectal cancer. And there, what we think is maybe happening is that, you know, the adenomas or these little outgrowths are pushing above the mucus layer and providing a landing patch for these biofilm producing organisms like Fusobacterium to land on, make a bacterial biofilm and stick around and produce more biomass. How aging in the oral microbiome affects gut microbiome composition, I doubt it's a big effect, but in certain cases like cancer, it perhaps matters.
Yeah, so it's not any kind of physiological changes that happen with aging, sudden change in dental health and related periods where oral flora is completely different, at least from, the perception. I don't know about the composition, and that doesn't really impact the trajectories of what could happen with gut microbiome changes and therefore health.
I don't think the oral's doing much to what's going on in the gut for the most part, but I do think that there's important things going on. So, for example, host amylase copy number. So we have different copy numbers of these genes to break down starch, and people who have high copy number, they tend to actually absorb more calories from starches, and they liberate more sugar in places like the mouth. And so dental caries can actually vary based on host genome variation because the microbes might have access to more sugar. But as far as what's going on in the gut, I don't think there's a huge effect going on there.
Okay, I do want to make sure we have time for a break in between sessions. So I want to thank all of the panelists today, and I want to thank the organizers for inviting us. I think this was a lot of fun, and I learned a ton. And I'm sure I speak for everyone where if we didn't get to a question, or if you have any other follow-up questions, please feel free to reach out to us. And we'll see you in the next session.
Started with the third and final session of this workshop. My name's Matt Rossman. I'll be chairing the session on special topics in precision nutrition for aging. The first speaker is Dr. John Batsis, who is a clinician and health services researcher with a joint appointment in the Division of Geriatric Medicine, as well as the Department of Nutrition at the University of North Carolina, Gilling School of Global Public Health. He attended medical school at the University of Dublin, Trinity College, and completed his residency and fellowship at the Mayo Clinic. His specific clinical and research interests include the synergistic impact of obesity, as well as low muscle mass and strength on important outcomes relevant to older adults, including physical function and mortality. And today he'll be talking about the importance of maintaining muscle health. So I'll hand it over to Dr. Batsis.
Great, thank you, Dr. Rossman. And can you see my slides okay? Great. Yeah. Well, first, thank you very much for the kind introduction and the invitation to speak here to the group today. Really would like to share a topic that's really passionate to me is really the treatment of obesity in older adults and how we really need to be thinking about maintaining muscle health as well. So, funding and some disclosures as well. So, just to start off with, and this is probably nothing new to this group, but older adults are really a heterogeneous group. And when we think about an older adult isn't one older adult is one older adult. None are the same and ever alike. And often a number of our treatment approaches are really focused on a one size fits all. So we take guidelines and we apply them, and we don't really think about the heterogeneity, both clinical and biological, that older adults are privy to. And as we know, many existing clinical trials often exclude older adult populations or include those that are very functionally capable in their trial design. And we really need to think about the importance of multiple different types of treatments when we think about treating obesity in this population. And obesity, as we all know, is heterogeneous as chronic disease itself. And I was privileged to be part of a National Council on Aging's Obesity and Equity Roundtable, which really emphasized the importance of personalizing obesity treatments, really thinking through comprehensive, both clinical and tailored approaches, and not only using lifestyle, but thinking about pharmacotherapy and surgery. So before I dive into the topic I've been asked to speak about, is we really want to think about changes in body composition that occur with the aging process. And many of you are I'm sure, familiar with this, where weight actually increases during the life cycle till about the 7th, 8th decade of life, and then starts tailing off. There's a parallel increases in fat mass, but we know that lean muscle mass actually peaks in the 3rd to 4th decade of life. And then subsequent to that, individuals, once they hit their early midlife, that amount of lean muscle mass actually diminishes to a certain point. And everybody's trajectory is different. And they differ slightly between male and female, so there's sex-specific differences as well that we need to be thinking about when we're tailoring different treatments. So this is some really formative data and really foundational data that I think is really critically important when we think about muscle mass, muscle strength. And just because you have low muscle mass does not necessarily mean you have low muscle strength, and vice versa. And this is actually seen that we see in the Health Aging Body Composition study, which demonstrated over a period of a 3-year period of time that actually muscle strength in those over the age of 70 actually demonstrated by BMI category, body mass index category, a reduction in strength over 3 years, while there was essentially a maintenance of lean mass. So strength actually may precede the reductions in lean mass that observed with aging. And then we also see this in terms of from a performance or functional measures. And this is a large study conducted by Stephanie Studensky a number of years ago, but really is pivotal, that demonstrates peak gait speed, which is a marker of physical function and a marker of overall health, peaks in the 3rd to 4th decade of life. And then you can see individuals have different downhill trajectories. So we need to be thinking about this when we're thinking about therapies for obesity. Now, we've often talked about obesity in both lay and scientific literature using body mass index, or BMI. And this is an earlier study that our group had conducted, really using some NHANES data, which really demonstrated the poor sensitivity of BMI in identifying body fat in those both males and females. And we actually demonstrated that the diagnostic accuracy actually diminished with increasing age. And a major reason for that is not only individuals shrink, not only individuals gain weight, but there's a redistribution of fat and muscle and a reduction in muscle with aging that BMI really does not capture. So we need to think about going beyond BMI in terms of the diagnosis of obesity. And this is actually really well captured in a consortium, the European Society for Parenteral and Enteral Nutrition and the European Association for the Study of Obesity actually have put out some consensus guidelines on how to identify those with obesity with sarcopenia, the loss of muscle mass, strength, and/or function with age. And BMI or waist circumference in conjunction with a clinical measure of sarcopenia is used just for screening, and then we need to be thinking about it differently using performance measures and body composition, formally measuring body composition for its diagnosis. So I put this here because it really puts into context in terms of what the ensuing slides that I'm going to be presenting. So this concept of obesity with sarcopenia or sarcopenic obesity, definitions have changed markedly over the last 2 to 3 decades. And really, we know from a series of epidemiological studies, a number of them that a number of citations here that I've been privileged to be part of, that having a diagnosis of obesity with sarcopenia, and this is defined a variety of different ways, low muscle mass, low muscle strength, both, leads to increased risk of functional impairments, increased risk for falls, increased mortality risk, reductions in quality of life, and actually there's emerging data that's suggesting that there's an increased risk for cognitive impairment and dysfunction. A key tenet to this is we can see from the MRI imaging on the right. So this MRI image is a cross-sectional image of one's quadricep muscle. And you can see hyperintensity on the panel on the left that indicates fat deposition within the muscle. So this myosteatosis is actually very pro-inflammatory and really leads to a vicious cycle in this geriatric syndrome, compared to those without obesity on the right, which you can see really defined degrees of the muscle architecture. This intramuscular adipose tissue, and again, using Health ABC study, they used CT scans to look at this. And there are really the gold standard is really CT or MRI imaging to be able to assess this. You can't look at this using DEXA or bioelectrical impedance analysis. But individuals in this study actually who were weight stable or that gained weight actually developed more intramuscular adipose tissue. Those that lost weight still gained that intramuscular adipose tissue, but much less so. So we know with the aging process, there is a natural tendency to deposit fat within the muscle. And this in itself leads to major issues from a underlying biological basis, but that then translate into clinical dysfunction. So the main issue here is we know that sarcopenia is bad. We know that sarcopenia with obesity is bad. And this is really has come full throttle, I think, and as I'll show you in the next coming slides, with the emergence of some of our novel pharmacotherapy. Is all weight loss the same? And I really want to hammer the point home here that all weight loss is not the same. When one loses weight, one loses both fat mass, but fat-free mass as well. So you lose muscle, bone, and organ tissue as well. And it's really unclear when you lose weight, how these changes in the reductions in fat-free mass, do they change function, fracture risk, and at what thresholds? What is the sweet spot? We don't know that question and the answer to that question. But I'd like to kind of take you back to one of the seminal lifestyle intervention studies that Dennis Villarreal conducted, published a number of years ago, but really true to the point, demonstrated a control group versus a aerobic and diet group, a resistant exercise and diet group, and a combination aerobic and resistant exercise with dietary modification group, a combination group. Here's the take home. He demonstrated in this study, and this is a cohort of older adults with obesity with frailty, that a lifestyle intervention that incorporated dietary modification, caloric restriction, changes in diet quality, aerobic exercise, resistant exercise, led to a synergistic improvement in physical performance. And he used the physical performance test and really demonstrated the sum of the parts is more than the whole. And you see that mean weight loss is 9%. This is really important because you lost weight, but individuals in the combination group actually did better functionally. And we also know from studies conducted, there's a systematic review and meta-analysis that Steve Krzyzewski and his group at Wake Forest conducted, that lifestyle interventions, these are all lifestyle interventions of older adults with obesity, that there was a 15% reduction in long-term risk and mortality. So we know that they work. We know that it improves function. We know that it actually improves risk of death, reduces the risk of death. Now, here's the problem, is when, as you recall, when one loses weight, one loses both fat and lean muscle mass. And Dr. Villarreal and his group really identified this. In this group, this is on the panel on the left is lean mass, on the panel on the right, strength. And you can see there was reductions in lean mass. So the mean weight loss was 9%, but those in the aerobic group lost considerable amount of lean muscle mass Those in the resistant exercise group, there was mitigation of the loss of muscle mass, and those in the combination group actually were kind of in between. And then when you look at strength changes, those in the resistance and the combination group actually improved more than either from baseline. So we know you're going to lose muscle mass, lean muscle mass, but the question is to what degree, and can you mitigate it? I think that is a key clinical question. He took it one step further, and he had actually conducted some MRI imaging. And what he found is that the combination group actually led to reductions in thigh intramuscular adipose tissue, more so than the other groups, and a reduction in abdominal visceral adipose tissue. So we know that these are highly metabolic tissues, very pro-inflammatory, lead to a vicious cycle of impairment affecting adipokines, cytokines, and other fat muscle mediators. But we know that this type of lifestyle intervention, the combination, actually can reduce both more synergistically. But what happens after weight loss, after an intervention? And Kristen Beavers from the Wake Forest group actually conducted this really interesting study where individuals who lost weight right to the point where the intervention, it was a 5-month randomized controlled trial in postmenopausal women. And you can see the old adage when we think about how much weight you lose is between 2/3 to 3/4 is fat, 1/4 to 2/3 is muscle or fat-free mass. The predominant amount is constituent is that of muscle. That said, post-intervention to a 6-month follow-up, individuals regained some of the fat mass, but they really gained minimal amount of muscle mass, and that actually worsened the longer out they were. So yes, you can lose muscle mass and you lose fat mass, but subsequent, when you stop the intervention, here's the problem. For every 1 kilogram of fat regained over the following year, only 0.12 kilograms of lean mass was regained. So your ratio and or your the type, potentially the muscle quality actually differs. And this is, again, a major issue because where is this fat deposited? It's actually often deposited in ectopic tissues and including muscle. We also saw this in the Look Ahead study. They observed changes in lean muscle mass at 8 years, and they found increases in lean muscle mass in the first year, but then by year 8, the intensive lifestyle intervention versus the control actually was similar. And the decline was really very similar and almost linear over the 8-year period of time. I share with you some of the data on bariatric surgery, and this is done deliberately because we know that after bariatric surgery, you lose both in the laparoscopic Roux-en-Y gastric bypass, you lose both fat and lean mass, but in the sleeve gastrectomy, you lose similar amounts, although albeit the amount of weight loss and the amount of lean muscle mass is less in this procedure. And this is really important because you can achieve profound weight loss with bariatric surgery. And I'd be remiss not to talk about obesity in older adults without talking about the new GLP-1 GIP medications. This is a study that looked using the SUSTAIN trials of semaglutide and really compared young versus old. Take-home point here is that the efficacy of semaglutide when they compared older adults versus younger adults was very similar across these trials. So really achieving similar amounts of weight loss. That said, there are very, very few. There's a huge research gap in understanding the changes in body composition resulting from these medications. And this is Wilding's initial semaglutide study, which demonstrated a reduction in total fat mass, but also a reduction in total lean body mass. So when you look at the average rate and the proportion, approximately 40% of the weight loss was lean body mass, and this is using DEXA. There's some other studies that I won't go into that looked at differences between different types of imaging modalities. They actually differ. You may actually get different types of either ratios or quantities of fat and lean body mass loss, depending on the imaging modality you use. And this is using tirzepatide, the SURMOUNT-1, which again demonstrated almost a 26% reduction in total fat mass loss, but an 8.3% reduction in lean mass loss. What about physical function in these medicines? Really important, because there are very few studies. Some of the more recent heart failure studies have looked at 6-minute walk tests, and that's pretty much the extent. But self-reported function, which is not as valid as objective measures of physical function, demonstrated some improvement, but we need data. We're critically lacking data here. Important just to be aware of, there are some activin 2 type receptor antibodies. Bimagrumab is the prototype right now. There are a number that are being investigated in phase 2 and phase 3 trials. But this is a promising medicine, which may actually be helpful in thinking about prevention of muscle loss during weight loss efforts, and it actually demonstrated some improvements in lean muscle mass loss. And the data is still pending here as whether or not this improves physical function. So in the remaining couple of minutes here, I want to just focus on future research efforts and gaps. It's really one of the challenges that we've had existing in obesity medicine is really how do we apply what we know into different healthcare delivery models, both with lifestyle medications and surgery. We need to think about the life cycle. What happens early on that affects distally? As a geriatrician, I see patients in my clinic at the end when they're at the tipping point or either succumbing to the complications of obesity. We need to be thinking and going beyond disease states and thinking about geriatric syndromes. And importantly, relevant to this symposium, thinking about heterogeneity of treatment response. I'll come back to that. We've heard a little bit about translational geroscience, thinking about how we can use technology to help us, and thinking about the safety of weight loss of muscle mass, loss of bone. And we have major research gaps on studies in incretin therapies amongst aging populations. You've seen a rendition of this figure from other speakers today, but we really need to be understanding the biology of obesity, multiple chronic conditions, and frailty, and how it leads to disability and death. And understanding this will allow us to then design clinical trials accordingly. One of the things that we need to be thinking about from a heterogeneity response is that not all individuals will respond to a given intervention. And these are the premise of using precision medicine trial designs, sequential multiple assignment randomized trials, factorial designs, and then using machine learning precision medicine analytics to identify dynamic treatment responses to identify what subgroups actually can benefit from intervention. And it may be that some individuals in the intervention arm don't respond. Some individuals in the usual care, control, or comparative arm don't respond. But there may be others in those other arms that do respond. So we want to be able to understand what are their baseline characteristics and how those can inform the tailored delivery of interventions. And then really thinking about phenotyping. We have at our source here multiple sources of data: clinical, biological, functional, and mobile health. How can we identify different phenotypes to identify in the diagnosis, management, treatment of these individuals? And really, I think we're really at the tipping point of understanding this in both obesity, sarcopenia, sarcopenic obesity, and aging as overall. And with that, I want to thank you very much for your time and your attention today.
Thank you. So if people have questions, if they could just enter them into the Q&A chat. I guess I can start with one. So in regard to, you presented some interesting data regarding the combination of aerobic and resistance training. Is there any thought to kind of combine pharmacological treatments such as the GLP-1 agonist with resistance training to offset the loss in muscle mass?
And that's really the $1 million question. It's a great, we're all thinking about it. The challenge really is it needs to be done in a pragmatic way. These medicines are so expensive, and they're access issues, insurance issues, geographic challenges. But you're absolutely right. You know, these, you really need to be, in my mind, I think we really need to be thinking these as layered. You need to start with a lifestyle. And then in those that are actually really, there are individuals who lifestyle works and they may be maintained, and the individuals that it doesn't work, or, you know, individuals aren't able to lose enough weight to improve their function, then the next layer really would be potentially pharmacotherapy and or surgery for that matter. So we need to be thinking about this as a continuum, not as, you know, different boxes, which I worry we are starting to think about that. Just here's your medicine and off you go. And that's really not the right approach, I think.
Great. Yeah, any other questions from, I have one question related to visceral fat. Does visceral fat increase or decrease with aging?
Great question. So visceral fat, actually, there is a redistribution of fat with aging, and it really, you know, it redistributes viscerally, which then ends up leading to more increased inflammatory mediators, frailty, and the like. And that's one of the things you want to try to avoid from a life cycle standpoint, is that potential redistribution, or to mitigate that, to reduce the risk of long-term outcomes or adverse outcomes.
Great. All right, well, thank you very much. I don't think we have any other questions in the chat, but I'm the next speaker, so I'll just start sharing my screen, my slides. Okay. So my name is Matt Rossman again. I'm from the University of Colorado Boulder, and today I'm going to be talking about dietary supplementation-based strategies for targeting hallmarks of vascular aging and improving endothelial function. Cardiovascular diseases are the leading cause of death in the United States, and aging is the primary non-modifiable risk factor for cardiovascular disease. A key contributor to the increased risk of cardiovascular disease with aging is the development of endothelial dysfunction.